are actually this fascinating mix of electrochemical interaction. So an electrical impulse will propagate down the axon, and it will cause vesicles in what's called the synaptic terminal to protrude into what's called the synaptic cleft. It releases neurotransmitters which diffuse across the synapse and bind onto receptors on the opposite side. These open up ion channels which cause currents to either cause the membrane potential of the downstream neuron to either increase or decrease. There's two types of interactions that can be mediated through many different currents, but the two types of interactions are either excitatory or inhibitory. So the way you can sort of simplify this whole picture is that a neuron either has an effect to excite another neuron or inhibit another neuron. And with these long, big trees of dendrites, branches of dendrites, they integrate all sorts of inputs from many, many, many different neurons. And all those inputs propagate down passively and actively down these long dendrites. And their effect summates in the soma, and once a threshold or a threshold-like point is reached, that action potential is generated and propagates. So this is thought to be the main form of communication between neurons. It's not necessarily clear that we know that's a fact. There may be other ways that neurons are truly communicating, and we've just decided to put a big flag on this. Okay? But this is one of the things we study quite a bit. So just to give you a clue, it's a big wiry mess of dendrites and axons interconnecting everywhere. It's a very complicated picture. So I'm going to talk about the visual cortex and results in the visual cortex. So this is the visual space, and this part of the visual space propagates into through the retina, down the optic nerve to the LGN, and then back here to the visual cortex. So the left part of the visual space lands on the right part of your visual cortex. And the right part of your visual space lands on the left part of your visual cortex. So your visual cortex is really two pieces. And this is the primary visual cortex.
Stefanos,
Yes.
your light is not working.
I know, it's got
so
a problem.
maybe you can close it and show it, okay? Because
Sorry?
maybe you can go closer to
Oh,
point
and
the
point
show,
to
yes,
it?
and show yes.
All right. Okay, so basically these two regions here. Okay, they're about the size of a credit card. And one property that they have, well, I should say, there's a wonderful map that maps the visual space onto the primary visual cortex. It's actually a complex logarithmic map, but a point here in the visual space actually maps onto a point on the visual cortex. So if I shine a light here, there would be a small region in the primary visual cortex that would be stimulated. And these neurons in this part of the brain respond to different features of the stimulus. For example, the direction in which it's moving. I'm going to talk specifically about orientation. So this is, for example, an oriented stimulus. It's an object that has an orientation to it. And a fascinating thing about many neurons in the visual cortex is they respond to the orientation that they are being presented with. So what you can do is shine light bars into, well, I should say each neuron has what's called a receptive field. So in that visual space, there's a tiny little window that each neuron sees, and they all overlap and cover the whole visual space. So if you place an oriented bar in that receptive field, depending on the orientation that you hold the bar, the neuron will fire action potentials. Okay, those spikes that propagate down the axon, it'll fire them at a different rate depending on the orientation. And in fact, these neurons are tuned to like a particular orientation, which is called the preferred orientation. So here, this particular neuron likes the vertical orientation, it fires at the highest rate at the vertical orientation, and as you rotate it away, the firing rate decreases until it's orthogonal, in which case it barely fires at all. So this is called orientation tuning. The response of these neurons is tuned for orientation. And one thing I'm going to talk about today is orientation selectivity. Selectivity specifically refers to how wide or narrow is this tuning curve. So the more selective it is, the more it prefers a smaller range of orientations. So this is a picture of the visual cortex. This is created by optical imaging the visual cortex, stimulating all the different neurons with different oriented bars and identifying which neurons, what their preferred orientation is, and how does that preferred orientation vary as a function of space across the visual cortex. And you can see it's got a nearly periodic structure to it, although it's obviously not perfect. But you have some really interesting behavior. There's domains where you move continuously through different orientations. So the color here represents the preferred orientation of that group of cells. So here they like this horizontal, here they like vertical, back to horizontal. There's domains where you move continuously through the orientations, but you also have these little pinwheel centers, and these actually form interesting singularities in what's called the orientation map. And those are actually a true pinwheel center. There's a group in Harvard that has used two-photon microscopy to actually determine that it really comes down to a point. It's not this blurry center, but it actually comes into a point. So when I talk about the response of these neurons to the orientation of the stimulus, I'm very much interested in how does this connect across space. So this kind of picture here will play a big role in the second part of my project as I'm looking at many different neurons across space interacting with each other. So I'm going to move now on to gamma oscillations. So this was the first topic. The second topic is about gamma oscillations, and intrinsic to that is synchrony. So just to give you some motivation, why do we even care about these things? Okay, we don't know what they're for. But they occur all over the place. They occur in the neocortex, which is the outer part of the brain. This is where your visual center is, where your motor center, where all your sensory cortices are, except for olfaction. The entorhinal cortex, the hippocampus, these are very much related to all sorts of spatial tasks and memory, the striatum, the olfactory bulb, the thalamus, all over the place these oscillations occur. There are differences between them in different places, but they come up all over the place. And it may be one of the basic mechanisms the brain uses to do things. What are those things? Yes?
What are these oscillations?
I'll show you some pictures. I was going to put the pictures first and then the motivation, and then
Okay.
So there's a causality issue here, I guess. So just right after this, I'll show you what they are. So, well, here, let me just show you what they are. So here are gamma oscillations. These are in the visual cortex. So oscillations have been studied since the 20s when we first started putting electrodes on the brain and measuring brain waves through EEG. But that's on the exterior of the brain. That's summating a lot of the electric fields from many, many different, generated by many, many different neurons. And so Charlie Gray and Wolf Singer were the first to really record inside of the brain and find a certain type of oscillation, the gamma oscillation that we see all over the place. So when you record extracellularly, you get a signal, and you have to filter that signal. If you filter it at very low frequencies, you get an oscillation that looks like this. There's a period in here that has a very rapid, regular oscillation. If you filter at high frequencies, you get what are these spikes. These are the action potentials. And what you can see here are many, many different action potentials. So if we look over a very small interval of time, here's the LFP. And you can think of the LFP as this low frequency, well, it's fairly high frequency, but a low frequency signal that's representing the output of many, many different neurons. And you have this oscillation. The oscillation is somewhere between 30 and 100 hertz, usually in the 30 to 50 hertz range. And if you filter at the high frequencies where you pick up the individual spikes of many different neurons, what you find is the spikes occur during certain phases of that oscillation, followed by periods of quiescence in between. So these vertical lines here are the spikes. Here's another example. So here's a long period of oscillations. If you blow that up and look inside, you see periods of spikes, quiescent periods of spikes. And it's about in the 30 to 50 hertz range. And if you look really closely in one of these little bursts here, you see action potentials. Now this is extracellular, but this was looked at intracellularly too, and all of this correlates. So here is based on the LFP. Here's the extracellular recordings, and here are the intracellular recordings. So a neuron is firing at those same points when you have the oscillations in the LFP and in the extracellular recordings. The thing about the extracellular recordings is you're recording many different neurons together. The intracellular, you're actually getting one single neuron and asking what is it doing. This particular one here is firing regularly. You don't always see that happen. Sometimes it's just one spike or two spikes. And it's really the population that actually fires in the oscillations, in these gamma oscillations. And the population is what's producing these oscillations. So many people have come up with all sorts of ideas about what are these oscillations used for. Okay? It could be to coordinate large ensembles of neurons to perform certain tasks. What are those tasks? There's all sorts of things they could be. It could be for coincidence detection. These oscillations could act to coordinate the behavior of neurons so that they're not firing and firing at certain times. They set up the ability for different neurons to synchronize with each other. And this could be used for coincidence detection. There's also a type of plasticity. Neurons in the brain rewire through their synapses. The synapses change. It's been well demonstrated that some types of the synaptic changes that you get, the plasticity that you get, depends on the spike timing. So if one neuron fires and then another neuron fires after it, the synapse can be strengthened. If that relationship is opposite, the synapse will be weakened. This can coordinate the timing of those spikes to help enhance that or prevent it. Gamma oscillations have also been found to be present during tasks where an animal is attending to an object. And so it could actually work in a few different ways here. One could be that the gamma oscillation is helping to focus in on one particular thing that needs to be attended to and ignore the other type of thing that you do not want to look at. It could be at a more rudimentary level, this idea here of sensory binding. There's this whole sense of if you are encoding something in the brain, like an object, right? If I told you about in the visual space, each neuron has a tiny little window that it sees. This object would span many of those different windows. So how does the brain take an object that it's seeing in little points and string it together and form one coherent object? How does the brain bind this object together? And it may be that it's during these oscillations forming cell assemblies that you could form this bound percept of. So there's many different things. Gamma oscillations are also present during working memory. This is the short-term memory that you have when you're trying to remember a location of something for a very short period of time, for example. So there are many different instances where this comes up. Input gain modulation has also been proposed to be facilitated by gamma oscillations. but no one knows. Okay, this is the thing. You can propose all these and create tons of experiments to really try to elucidate what are the roles of these oscillations. Yeah.
Over what length scale are these seen? Is it very localized or is it global?
It can be very long range. It's often short range. Okay, so that's one thing that happens in the visual cortex that I see, and I'll talk about. So it could be that these oscillations are coordinating localized structures. So for example, localized features in the visual space. And when you form cell assemblies, they could be helping to encode that. Now, I keep saying gamma oscillations and making them seem like they're responsible for all this. I would argue that's probably not what's going on, and many people don't think they are the thing that's responsible. What it could be is it could be creating the framework that could be used to do all these tasks. Because of this oscillatory framework that facilitates synchronization, that facilitates timing, that creates little windows when the neurons can fire, and having many different tasks being able to be performed in different windows, and coordinating these windows. So in a way, these oscillations could be more for coordinating things rather than performing the task itself. And so, but nobody knows. So this is why I scale back from saying, what do these things do? And I want to understand, what is the mechanism behind the phenomenon that we're seeing? And hopefully through that mechanism, we might be able to understand what is done. At the very minimum, it gives you a basic set of knowledge that you could use when addressing further questions. So, okay, I'm probably going very slowly through this, so I'll try to speed up. So it also is present in many different diseases, whether it is caused, so there's reductions in gamma oscillations in certain diseases, and this may be responsible for deficits in cognitive ability. In certain cases, it's enhanced. So it may be playing an important role in some of the pathologies and disorders. And so it's another important reason to study them. One thing that Gray and Singer found was that synchrony correlates with orientation preference. So when two neurons have a similar orientation preference, and they're firing frequently because they're stimulated by the preferred orientation, the gamma oscillation essentially coordinates that synchronization. So I don't have a picture for you there, but I'm going to demonstrate that in terms of my results. So here is a set of experimental results that my collaborators found. I did a lot of the data processing on this because they found a number of results, and I wanted to demonstrate that I could see all of those results in the basic data. And I wanted to imitate the modeling that I was going to do based on the experimental results. So I wanted to have a match between the two. So one of the first things that they did, and they published this in 2008, they did a functional connectivity analysis through a particular model. I'm not going to go into the details here, but what they found was that there seemed to be certain types of neurons, certain types of neurons that would synchronize more strongly than other neurons. And one of the features that these particular neurons had was they tended to have sharp, narrow tuning curves. So there was an interesting correlation that they noted there. They just happened to note it when they were looking at pictures like this. But what they found was that there was a subset of neurons that synchronized more strongly than others. And what we wanted to do is understand better what can we say about these particular neurons. So we've called them synchronization hubs because they synchronized strongly, they were able to make not anatomical connections. Okay, when we typically think of connections, we think of anatomical connections. These are connections through synchronization, meaning they're able to time their spikes very well with many other neurons compared with others. So for So this picture is showing a whole group of neurons here, and only a few of them are able to synchronize well with neurons in this other group here, whereas all these others do not. So they identified this group. And we wanted to look into more of what properties they had to try to see what might be the source of this. So this is the problem I want to focus in on. So in the presence of these gamma oscillations, what makes this particular group of cells synchronize better with other neurons? And I want to try to take into account all the data we see. So I spoke to them because they hadn't published this. And I wanted to understand better what is it that they could see in the data. And one thing that kept coming up was this idea that there was a relationship between synchronization, oscillations, and orientation selectivity. And I had been studying gamma oscillations for a while, and I had all these ideas about it. And the picture just started to come together for me in this discussion. So let me show you some of the basic results that you can see. So these are parallel recordings. So this is a probe that's got 16 different electrodes on it. It's being stuck orthogonally into the visual cortex. So you're seeing many different layers of the cortex, probably 3 layers, and then across, laterally across the cortex. So this would be within one of those orientation pinwheel type distances that you saw in the picture before. So this isn't spread across the visual cortex. This is in a very small, what we call a cortical column of the visual cortex, where all the orientations are represented. So I'm showing here rastergrams of these different, of the recordings from these different electrodes. So each one of these, what says CH, that's for channel, that corresponds to the number that's over here. And these recordings are the spikes in parallel of, well, a subset of the 16 units. And what you see is all across the probe, you see this strong synchronization. Some of them are tight, some of them are more broad. The difference between the dark spikes and the light spikes, so the dark spikes are individual neurons. The light spikes are what we call multi-unit recordings, which are really, it's the extracellular recording that's sampling many different neurons. So you're seeing here the spikes of many different neurons firing in the same channel. So you can think of the black ones as an individual unit or an individual neuron, and the light ones as many different neurons in that local region around the electrode. So these are 2 different instances here. The cat is being shown a sinusoidal grading at a different orientation, and so you're seeing the response to that orientation. So this is a grading, so it's light, dark, and it's moving across the receptive field. As it moves across, it stimulates that neuron. And instead of firing continuously, it fires in bursts that are basically at the frequency of gamma. So you see these windows where it fires, and then windows of quiet, windows where it fires, windows where it's quiet. And that's the gamma oscillation. But there's a variability across this. This one here fires continuously, and it's got small units. So you see a lot of it. I've pulled out a lot of the units that are that have more tight packets of spikes, because these are the ones that are like synchronization hubs. This channel 8 here, that unit is what I would call a synchronization hub. It's got a lot of spikes followed by a long period of quiescence. So how do we analyze all this? There's tons of data. We run many different trials. There's just too much data to really look at individual spikes in these rastergrams. So the rastergrams are nice because they show you very specific instances of what's going on, but we want to look over long periods of time and try to get a sense of how much synchrony is there really, what is the strength of the oscillations that we're seeing. So, so we can do this through what are called cross-correlograms or autocorrelograms. Yes.
How long is the visual stimulus present for?
Well, 5 seconds.
Okay.
And most of this recording that we're looking at and analyzing is over 2.5 seconds.
Okay.
And then run many, many different trials, and they're all randomized, and then we're looking at average data. That's not average data, but all the a lot of the data will be averaged over trials.
So basically, you're looking at the geometry of neural feedback, kind of like a structure of the echo in the system.
Right.
So it's not strictly feed forward. Okay, got it. Thanks.
So, okay, so I just tried to make a quick animation because I'm going to talk about these cross-correlograms and autocorrelograms. So how are they formed? Basically, if we can look at 2 spike trains. So this is just whenever a neuron fires a spike or a unit fires a spike, you mark a line at a certain point in time, and you can... So instead of looking at the entire voltage trace, you're just looking at points in time where there's an action potential. And then the question is, how coordinated are these action potentials? So an autocorrelogram or a cross-correlogram looks at 2 different things. A cross-correlogram looks at one spike train relative to another. An autocorrelogram looks at itself. These are histograms of spike timing. So to start out with a cross-correlogram, here's one spike in one neuron. There's no spike coinciding with it right here. Okay, but to make this histogram is to say, what is the time difference? What's the lag between this spike, this and this one, this and this one, this and this one? And we can mark those lags in terms of bins, because these spikes are infinitesimally thin. So they're at these tiny little points. So what we're doing is breaking the full set of time into bins and saying, in this bin, is there another spike? This one's close enough to this that both of them lie in this bin, so we'll mark a time. And that marks the lag between the 2 spikes. So you could say synchrony, to a mathematician might mean that there's 0 difference in the timing. But in the brain, that's not necessarily true, because the way synapses work is over many milliseconds of time. So really, synchrony could actually be over a larger period of time. I bin this stuff in terms of 1 to 3 millisecond bins. And so when you start coordinating these, you can start counting. So there's 2 spikes here, 2, 1 in all of these. And you do this for every pair of spikes to see what is the time difference between these. And this is after 1. Then you move on to the next one and add more. And you do this for every spike train, and you can average these over trials, and you can get a sense how often so when you look here at the point 0, how often are 2 spikes in 2 spike trains almost coinciding, and how often are they at some lag, coinciding with some lag? Now, why is the lag important? Because if something's oscillating, then that means, if it were perfectly periodic, that would mean after some period of time, then you would see it coinciding again. As you shift to signs relative to each other, there's going to be a point where they re-overlap. The problem is, these are not perfectly periodic oscillations. They fluctuate from cycle to cycle. But still, if they're oscillations, then you're going to see peaks that come out. So you can use the cross-correlogram and autocorrelogram to look for oscillations, synchrony, co-oscillations between neurons. And so to get a sense just visually of how strong an oscillation is, we do something that's more precise as far as calculation, but just so that you can look at a cross-parallelogram and get a sense. The amplitude of the central peak versus the offset, that ratio would be a good measure for the estimate of the strength of synchrony. Meaning, if this were completely flat, then no spike is really coinciding any more than at any other lag. But if this starts peaking, that means there are many more pairs of spikes that are coinciding if you look across the data. Okay? And if you start seeing peaks here on the side, the satellite peaks, then that means as you're shifting these two spike trains, the spikes are coinciding after a predictable amount of time. And that should correspond to the period, approximate period of the oscillation that we're seeing, the gamma oscillation. Okay, so then what you can say is the strength of that oscillation would correspond to how tall this peak is, or how tall this peak and this peak is. You could look at all sorts of... This is just a way to estimate so you can look at this and get an immediate sense. You really want to do this with a better calculation if you're going to actually do this precisely. But for example, here are two units that are, I would call synchronization hubs. You notice they have a narrow tuning curve. This is their autocorrellogram, so that's the spike train against itself. Okay, you would expect a tall peak in the center because that corresponds to them coinciding. Now these are not single units, but they're multi-units, so there are many different spikes. But you see, there aren't many spikes coinciding after some lag, and then they rise again. So this is a neuron that or a group of neurons that have a very strong oscillation going on. And if you look between the two neurons, this is the cross-parallelogram, that's looking at the pairings of the spike trains from this one and this one. What this shows is that these two neurons are coinciding quite a bit. If you shift them, they're not coinciding, and then they start coinciding again. What this means here, this here means there is a roughly periodic oscillation, and it's seeing itself as this spike train is shifted against itself, it's seeing its own oscillation. But between this one and this one getting this, that means the two oscillations between these two neurons are very similar and very much in phase. And I showed you the picture before of the spike trains, the rastograms, and you saw how lined up all the spikes were in those columns. Okay? That is what you're seeing here. You're seeing the synchrony, but then you're seeing that they're pretty much in phase across the whole probe. So this is an important part of my story, is that the fact that they're in phase everywhere is a strong indicator that something is coordinating them all on the, at least on the spatial scale of the probe. Okay. And so this is part of what I put in my model. So this is a little bit of a complicated picture, but here I'm just giving you examples of 4 different units. And these are 2 units that like the same orientation. These have different orientations. So here's the orientation tuning curve. So this shows the neuron fires more strongly at these 2 orientations and weakly at these 2 orientations. Similarly here, these 2 have a similar orientation. These 2 have a similar orientation. These 2 have strong oscillations here in their autocorrellograms. And what you see is between them, they also have a very tall central peak, meaning these 2 neurons are very synchronized. And they oscillate very similarly with each other. These are what I would call non-synchronization hubs. And it doesn't mean that they're not synchronizing. They don't synchronize nearly as strongly as these others. So one thing this picture shows you is that the synchronization hubs, these 2, synchronize well with themselves, as well as the other neurons. But the two non-synchronization hubs here synchronize the most weakly, and their co-oscillations are the weakest. So this is a trend that I want you to notice, because this is a trend I want to be able to reproduce in a very simple way. So this is for one stimulus orientation. If that stimulus orientation varies from 150 degrees to 180 to 210, okay, the picture doesn't change too dramatically. It does a little bit, but what you see across this is the synchronization hubs maintain their oscillations, they maintain their synchrony. And this picture here, these three plots, are plotting the strength of synchrony between all pairings. The tallest one here is always between the two synchronization hubs. The shortest one here is always between the two non-synchronization hubs. And that comes out in every case. This will vary quite a bit, but it always comes out in that same order. So that is what you would call an anecdote. That's just a couple different units. That's to give you an idea. But what we really want to be able to do is look at this all across all the units that we're measuring. And so what we find is there's a correlation between the three properties that I told you about. The strength of the oscillations of the units, the sharpness of their tuning curves, and the strength with which they synchronize with other neurons. And so this first plot here, so this shows what we call a synchronization hub. It's got a very sharp tuning curve. It's got a tall central peak, which we expect because this is an autoparallelogram. We have it of itself. But what this picture is supposed to show you is it's strongly oscillating. Here, this is a non-synchronization hub. It's got a very broad tuning curve, and the oscillations are very weak. Those are what you call the poster childs for these two different types. Really, there's a spectrum going from strongest to weakest. So don't think there's really two types. There's really a spectrum, but we're using the two types to just contrast two ends of the spectrum, really. So here, what you see is a correlation between the oscillation strength and the orientation selectivity, the synchrony strength and the orientation selectivity, the synchrony strength and the oscillation strength. That's for one cat. This is for another cat. You see a very similar behavior. And so what we're claiming is the synchronization hubs are essentially this group that lies up here. They're the ones that have the strongest oscillations, the strongest synchrony. So we call them promiscuous synchronizers because they're less discriminant about who they synchronize with. The non-synchronization hubs do synchronize, but they synchronize less frequently and with fewer different neurons. And they tend to be the synchronization hubs that they synchronize with. Okay, so another feature is this idea of that synchronization hubs maintain strong oscillations across different stimulus orientations. So here, these are, again, the cross-correlograms between these different units. Here are the autocorrelograms. So these are where you can see the oscillation strength of the synchronization hub. So I would call these a sort of decrease along the spectrum of these set units. So this would be a synchronization hub, this would be a non-synchronization hub. And what you find is as you change the orientation, okay, here, this neuron stops oscillating and stops synchronizing with everybody else. The synchronization hub maintains the strongest oscillations and the strongest synchrony. So, and again, you can look at this across all the different units. And what we did here was just ask the ratio, look at, plot one thing against the other. So there's the oscillation strength of the preferred oscillation, and then the oscillation, the minimum value of the oscillation strength at the two neighboring orientations and asking, is there a correlation between those? And in both cases there is, and what this indicates is as you move away from the preferred orientation, the synchronization hubs, which are these outwards, maintain the strongest oscillations, and everyone else decreases. So you asked about longer range. Okay. So this is so when I looked across two probes, which could be spaced a millimeter or more apart, okay, there is very little synchrony. Now there can be at many times, and this has been found before, but what I find is that the ones who do synchronize well are the synchronization hubs. They're the ones that are capable. So this is, and what this shows here, okay, so this is one probe, the green is one probe, the blue is another probe, and I'm showing you two different units in each probe. So this here is the cross-parallelogram between the units in one probe. This is the cross-parallelogram between the units in the other probe. And these four are the cross-parallelograms across the probe. So this particular orientation here, there's strong oscillations and strong synchrony amongst all of them. Okay, it's a little bit weaker here between the two probes. But what you see as you vary the orientation, the oscillations here still stay strong within one probe. But the oscillations and synchrony weaken between the two probes. And if you look, why would you expect this? Synchrony and co-oscillation is decreasing. Okay, a natural way that's going to come up is if the two oscillations are no longer in phase with each other. You're going to lose synchrony because the spikes are not lining up anymore, and you're going to lose the co-oscillation because the phases are different. Okay? And when you look at rastograms, I've looked at tons of these over different periods of time. This would be for this first stimulus orientation. And what I've done here is I'm trying to plot a whole bunch of different things so that you see them. The dark lines are the spikes in each unit. What I've done is I've extended those spikes so it goes and shows you what is the probe doing. So you're looking at each unit on the probe and then the full probe. So you can think, this is the gamma oscillation for one probe. You can see the periods. Each unit's firing in a different way, but they're all firing roughly in the same window. And this is for the other probe. And you can see these windows of firing are lining up, okay? Not always, but predominantly across. So this is going to give you what you would expect, which is strong synchrony between the two probes, as well as the co-oscillation. But when you go to this stimulus orientation here, you see that starts to degrade quite a bit. Okay, so my modeling goal, and I'll try to get through this, is to identify a mechanism that can account for the correlation between the strength of synchrony, the strength of oscillations, and orientation selectivity. Okay, now I'm going to mostly talk about these two. And this is something I technically need to do in a bigger model, but I touch on at this level. Okay, so there is a heterogeneity of these features across neurons in the primary visual cortex. And what we're calling the synchronization hubs, okay, we're calling them the neurons that synchronize the strongest, they oscillate the strongest, and they tend to have the sharpest tuning curves. They don't need to have the sharpest tuning curves, but they tend to. So one big question is, how do gamma oscillations arise? So I want to give you some basics about gamma oscillations. You might think that there's some remote pacemaker that's impinging on many different neurons and causing them to fire. Now there would be some delay if that were to happen, but if one neuron somewhere in the brain was connecting with a whole bunch of different neurons, it could coordinate all of them. Okay, you might have a local pacemaker that's doing the very same thing. Right? We know that this is what happens in the heart. There is a pacemaker that generates the normal functioning of the heart. But that doesn't seem to be the case at all in these oscillations. Many, many different studies have essentially come to the conclusion that this is a network phenomenon. A phenomenon that emerges out of the interactions of the whole network, not by one pacemaker. Now, in the visual cortex, it's a special place where there are cells that do fire at frequency of gamma. And so they may actually be acting in some way as a pacemaker, but this is not 100% clear. But as I told you, gamma oscillations occur all over the place where there aren't such neurons. So, one big question is what neurons are involved in generating the gamma oscillation. For a long time, people think of synchrony and oscillation in terms of excitatory neurons. If you want to synchronize with someone else, you want to cause someone else to fire nearly at the time when you fire. So if someone else is near firing and you're excitatory and you generate an action potential because you fire, and if you can cause someone else to fire immediately after, that can be synchrony. But these oscillations appear to be coming from a different mechanism, which is based on inhibition. And inhibition may make you wonder, how can inhibition cause synchrony and oscillations? So the way is, if you think of one inhibitory neuron firing regularly, it's going to cause another neuron not to fire at all. Because every time it fires, it holds this neuron and pulls it away from firing. But let's say both of these are firing. Then what inhibition does is it holds one neuron back a little bit because it inhibits it. And the other neuron will do the same to the first one. And they delay each other. And what you end up getting is that there's a perfect moment when both of them can fire. And that's right before the other one fires. They hold each other back, but what you have to understand about inhibition is it decays. So when an inhibitory neuron fires, it causes a current that has a roughly exponential decay. So as that current decays, there's a moment where that inhibition's the weakest, and another neuron can fire. The minute that other neuron fires, it will inhibit the other one. And this actually creates a stable attractor, where both neurons can fire just before each other fires, and get in a stable rhythm. Now, it turns out that, and that the oscillation frequency is determined by this decay time. So this is a feature that you can test. So how do we know all this happens? Well, two main sources have been used. One is in vitro models, so taking slices of the brain and doing all sorts of pharmacological experiments to look at different synaptic interactions, remove them, test all the possibilities to see what is necessary to maintain these rhythms, what blocks them, and you can uncover quite a bit of information. Someone might argue, well, slices aren't the real thing. Okay, yes, but we can start to understand how things work with slices and move upwards. Mathematical analysis and modeling has played a huge role in elucidating this. So really there's been a mix of slices and math models that have had a strong synergistic relationship to form a deep understanding of how these oscillations can arise. There's also lots of information from in vivo recordings that match a lot of what we understand about the in vitro models. And perhaps one of the coolest things that we have now is this idea of optogenetics, where we can essentially embed light-sensitive channels into neurons and activate them with light. So you can selectively choose certain neurons. So there's a particular inhibitory neuron that is believed to be responsible for these oscillations. You can embed these channels in different types of neurons and selectively activate those neurons and test whether that strengthens these oscillations or weakens them. And what you find is inhibitory neurons, when you activate them either in random ways or in synchronous ways, or in oscillatory ways with the same frequency as gamma, these cause the power of the oscillations to increase. So this is a really neat technique that you can even use to activate a single neuron with like 2-photon microscopy. It's really a really amazing new direction that's been going on for the past 8 years. Okay, it's not just the inhibitory neurons that are active here. It's actually a mix of excitatory and inhibitory neurons. And so it's really this complicated network effect, and that's for a whole other talk. One of the things that you find in vivo, so this is in vivo recordings, and looking at the LFP of the gamma oscillation, so this is a gamma oscillation in terms of the LFP, the local field potential, which is this low frequency filtered signal. So when you look at, so these are the inhibitory neurons here, these are excitatory neurons. What you see here is that the inhibitory neurons fire right at this phase in the oscillation, right at the negative peak, and the excitatory neurons fire just before it. So there's this phase relationship between the two. And what this you can see is, okay, the inhibitory neurons, this is a manifestation of the inhibitory neurons essentially shaping the firing window of the excitatory neurons. Okay, the excitatory neurons can fire up until when the inhibitory neurons quiet everything. So the inhibitory neurons, when we talk about those gamma oscillations, and they're large windows of quiescence, it's these inhibitory neurons that are firing that are forcing the quiescence in between the periods. So you can visualize this as that window of firing, and then this as the quiescence that ensues thereafter. So how do we model all this stuff? This is, again, a whole other topic, but I use two different types of models. One are what are called Hodgkin-Huxley type or conductance-based models, which are these nonlinear dynamical systems that govern the transmembrane potential. They give you action potentials, they give you all sorts of different types of currents that are responsible for different types of aspects. The problem with these models, while they're beautiful and they are more realistic, is they're computationally very intensive. And I'm looking at very long periods of time to build up these oscillations and to gain data. And what makes them computationally intensive is this action potential. Okay, that action potential requires very fine time steps to resolve. So I use an alternative. I use both because I want to know that both work. But an alternative is what is called the integrate and fire model, which is a model that tries to capture all of this subthreshold behavior here, but because the action potential is similar, from action potential to action potential, the part that's computationally intensive is tracing out this whole bit. So you can just ignore it, cut it off. And you might ask, well, what are you losing? Okay, that's something you should explore. You should understand what is the difference between the two. What I find is that in a lot of what I've done here, where I'm not trying to go after something very specific, but over a general behavior, I find the difference is almost negligible. If I cared about very specific spikes at very specific times, it would matter. But when it comes to the statistics that I'm looking at, I get roughly the same results for both. I'm glad that I do both so I can make that statement. But I can do far more with the integrate and fire because it's computationally significantly less intensive. So let me give you a quick idea of the model. So I want to build an entire network that does this, but that's doing way too much all at once. And so what I want to do is come up with a very simple idea, show that it can work, and then embed that into a big model. So I'm going to just try to show you what the simple idea is, and then show you that it can work in a very simple model. So the idea is that what's governing everything is inhibition. It's the strength of inhibition. And it's the strength of rhythmic inhibition. So, as I tried to argue, it's the inhibitory neurons that are predominantly responsible for the gamma oscillation. There's a mountain of evidence for this. So, you can imagine there's a pool of inhibitory neurons. Okay, really there's sort of 4 excitatory neurons to every inhibitory neuron. So there are really more excitatory neurons. But there's a pool of inhibitory neurons that are synchronizing with themselves and coordinating this oscillation. And the way I envision the difference between the synchronization hub and the non-synchronization hub is the strength of the inhibition that they receive. The synchronization hub receives stronger inhibition than the non-synchronization. But they fire at similar firing rates, so there's got to be a balance of excitation with that increase in inhibition. But this is one of the, a predominant rule in the brain is that there tends to be this balance of excitation and inhibition. But with that balance of excitation and yet an overall increase in inhibition, I can explain, at least in a plausible way, a number of these results. Whether this is the real thing or not is another question. So, this is just a cartoon that explains it. So, how do you get the oscillations? How do you get the synchrony? Well, I showed you those rastergrams from the beginning and showed you how all the spikes are lining up. So what this top part here indicates is you can think of these as, this isn't the current, but it's the conductance. Current is the conductance multiplied by the current, but you can think of the conductance as the current turning on and decaying. But what this is really is the summated. So this is the conductance from all of these different inhibitory neurons. And the brown is for the synchronization hub, and it gives you a larger amplitude than the yellow. So what that does, okay, I think I skipped over a slide. Anyway, so what I'm doing here, I'm really biting off more than I can chew with this talk. Tell you all this, but the way you should sort of envision this is I'm going to look at a bunch of different neurons, these here, and what they're going to receive is random excitatory inputs. Excitatory inputs are on a much shorter time scale than the inhibitory inputs. Okay, so if the inhibition lasts for 20 to 40 seconds, the excitatory milliseconds, the excitatory inputs last more on the order of 1 to 4 milliseconds. So it's much, much shorter. So you can imagine these neurons getting bombarded by random inputs. They may not be random, but I'm going to treat them as random to produce the images that we saw before. They may not be random, and this would still work out, and it might be more enhanced. But the idea is that the inhibitory neurons basically fire in an almost synchronous way, and they cause the probability of the excitatory neurons of firing to decrease during that period of time. As the inhibition wanes, the probability of the excitatory neurons firing increases, and that probability rises up to its peak just before the inhibitory neurons fire again. So this gray curve here, it's a cartoon for the probability of firing, is that window that you see. The neuron could fire somewhere in there. It doesn't have to fire. But if it fires, the likelihood of firing is greater here than anywhere else. Now the light blue curve is the non-synchronization hub. Because the inhibition isn't as strong early on, then it has a higher likelihood of firing. Now you might ask, why doesn't it go as high as the other one? Well, these two neurons fire at roughly the same firing rate. So you can think that the number of spikes each one produces is distributed across each cycle. So if they fire roughly the same amount, and the synchronization hubs are more strongly inhibited, that probability has to go somewhere, and it goes into that peak. Right? If there were no oscillation, then both of these would be the flat, same curve, and they'd have an equal probability of firing at any point. So what stronger inhibition is doing is causing the window for the synchronization hubs to become narrower, and increasing the likelihood of them firing closer to the end of the oscillation. So what this does is it allows them to be tighter knit in terms of when they fire with other neurons. So here, what you see, if the oscillations are perfectly coordinated from unit to unit, then these neurons fire, okay, with their highest probability at the same point where it's the highest probability that a non-synchronization hub is going to fire. Okay? So these may fire over here, but the non-synchronization hubs are most likely to fire precisely when the others are most likely to fire. This is what causes them to be synchronization hubs. Now you may ask, well, what if they're not as stimulated? Okay. Well, everything gets scaled down, so this result is still true. The highest likelihood of them firing is coordinated exactly with when the highest likelihood of the other are firing, and they don't fire as well here. So if synchronization hubs essentially distribute more of their spikes towards the end of the cycle, then they have a higher chance of synchronizing with somebody else. What can disrupt this? Well, a main thing would be if you start changing the phase relationship. Okay, so I'm arguing that that phase relationship causes us to be very sensitive, okay, for synchrony. If the phase relationship is disrupted, you will lose synchrony. And it can be disrupted just a little. Now, these oscillations are not perfectly periodic. Okay? Synchrony doesn't need them to be periodic. Synchrony just needs that during one period, you need this coordination. Okay? You could change the length of your period from cycle to cycle. All that matters is what happens during one cycle. Each cycle. So you need each cycle to be highly coordinated in terms of the probability of firing. These windows need to line up precisely, and they need to do that frequently. So this could happen here, where the windows shift a bit. But on average, they need to line up quite a bit to produce a lot of synchrony, or you're not going to get it. Okay, so this is the slide I was going to say. So these are, I have two different types of stochastic excitatory inputs, and the inhibition is stochastic in the sense that the length of the window changes from cycle to cycle. So I'm going to run through this. So, okay, I don't know if I'm going to be able to do this.
5 minutes.
5 minutes? Okay. So here, this shows you that the strength of inhibition can create that window and cause the spikes to pile up. So what I'm doing here is taking very, very long simulations. I know exactly when each cycle is. The cycle has different lengths, but I'm scaling them and overlapping them and showing you essentially that that inhibition does exactly what that cartoon picture does. So this shows you a strong accumulation of spikes in the neurons that receive the strongest inhibition near the end of the cycle, and here they're more almost uniformly distributed. This green here represents the case where there's no rhythmic inhibition shaping, okay, the firing, and it's essentially a neuron could fire at any point with equal probability. Okay, so if you look at cross-parallelograms, so here are 4 different units receiving different strength of inhibition, firing at the same firing rate. So here the inhibition is very weak, very small oscillations, very small central peak. The stronger the inhibition gets, the taller the central peak, the more likely they are to synchronize, the stronger the co-oscillation between them, and the stronger the oscillations in their autoparallelogram. So this is the increasing strength of inhibition. Here you can see the effect of the increasing strength of inhibition. It shapes the windows of firing so they become tighter. And so if this is a neuron that fires and it distributes its spikes across the whole cycle, okay, it's still going to be a little bit more likely to fire over here, which is when these guys want to fire. And so this increases the ability to synchronize. You get the same behavior and the coincidence or the synchrony strength of these different units. You get the strongest inhibition always generating the strongest synchrony. This is something I wish I had time to go into, but this essentially, in a sense, demonstrates why this idea works. It puts together 2 pieces that you need to argue why does just the idea work, but I don't have time to go into it, unfortunately. Here, this shows you if I have 2 strongly inhibited and 2 weakly inhibited, I get that same gradient. The strength of oscillations, the strength of synchrony is always highest between 2 synchronization hubs, always weakest between 2 non-synchronization hubs. It produces that same gradient in synchrony that you see in the experimental results. It just comes out naturally because of the strength of inhibition. Okay? If the coherence to 2 different neurons is very strong, and I have 2 other neurons where the coherence is strong, but then I disrupt the coherence between the 2 pairs, as you would naturally expect, the oscillations and the synchrony vanish. Okay, so you can have locally a very strong oscillation and synchrony, but you don't have coordination between 2. So here's, you could have a local group of neurons behaving in a certain way. They can synchronize well with another group, and then desynchronize. And that can change as a function of time. Many different groups of neurons could do this together at different points in time. And this was just a really quick picture as far as orientation tuning. I can get a similar tuning curve, both with strong and weak rhythmic inhibition. And then if I increase the strength of inhibition, I can sharpen the tuning curve and get strong oscillations. And so this is a correlation between the 2 that I would like to show in a full model, where the full model produces orientation tuning. So I just want to quickly acknowledge my collaborators. So this is work I did at the University of Pittsburgh as a postdoc with my mentor, John Rubin, Danko Nikolic, and Shan Yu. Were at the Max Planck Institute in Germany. They did the experimental results. And Abigail Snyder was a RU student of mine who worked with me on this project as an undergrad. I'd like to thank the National Science Foundation for funding these organizations for my co-authors. And this was recently accepted in May and published if you are interested in the reference. So just really quickly, future directions. I did this in a very simple model to show that it could work. And the next step is to build an entire network model that produces the gamma oscillations, that produces the orientation tuning, and behaves like this. All through a very simple, plausible mechanism of the strength of inhibition, which can be easily embedded into the model and doesn't require anything special. So you don't want a delicate mechanism that if something's slightly off, it won't work. It's got to be just right. This is a very robust mechanism that can be embedded in a variety of models, and it should work. I believe. So that's what I want to test out next.
Thank you.
Questions?
Oscillation strength. Did you look at more carefully with the autocorrelations at different lags and so forth?
Yes, for what specifically?
I mean, did you examine those things?
Yes, at different lags. So
What did you learn? I mean, are they significant, or what did you learn from it?
well, I think at different lags what you get is that that's where a lot of the effect of stronger oscillations come. Okay, so when you see those satellite peaks at different lags, that the problem with the satellite peaks is these oscillations are not periodic. Okay, so that's going to erode the satellite peaks because satellite peaks will appear more strongly if the oscillations are more periodic. So if they're if they're not very periodic, then this will erode them. So I would say when you see these strong, tall satellite peaks at different lags, then this is a really robust oscillation between these two different neurons, or in the autocorrelograms between itself. And so that indicates that the period of the oscillation is a bit more regular, and it's tighter. But the lags, what we didn't see a lot of was shifted peaks at different lags from the center. So what that indicated very strongly was that there was a lot of synchrony and not phase lags between one neuron and another neuron. Now that can happen at longer distances, but within one probe and even between the two probes, there wasn't much of a shift of the central peaks. So it wasn't that one was firing, say, before the other, but these were really strongly synchronized with zero lag. So that was a very predominant.
So a very similar question follow-up. So embedding this really simple model into a larger network, what kind of a network topology are you going to test or presume? Because this is kind of looking at very local connectivity versus a few non-local connections, I guess.
Yes. So I'm going to test out a few different models. I don't want to create too big of a model, but when I say network, I want to include many different neurons sufficient to produce, for example, gamma oscillations. Mm-hmm. So that could be a large, very local network that's only within one column. So different orientations are produced. Mm-hmm. So I'd like to produce a model that's based on a single column that has different orientations represented and a sufficient number of cells to produce the oscillations and the orientation too. And so how big of a model, that's a good question. But the topology is relatively well understood because of the anatomical connections between these neurons. We have tracer data that shows how they're connected to each other. And so I can do within one column, it's clear how to build that structure. And they're more strongly connected if they like the same, in a uniform way. Yeah.
Your graphs remind me of like a moire pattern, only in 4 dimensions. Sort of like two energy thistles merging, and then you're finding all of those points as they merge and demerge, as the surfaces come together and go apart. It's sort of like the National Science Foundation, it's like a synchronization hub, putting two of those together, and as the surfaces interact, that's what you're seeing as the synchronization. Is that making? I mean, this is what the moire thing in 4 dimensions is what your stuff is making me make sense out of it.
I'll have to think about it.
No questions? Thank you.
Just if there's anybody in the audience who would like to give a talk next semester, please. Or you have somebody,
Yes, yes.
or you have somebody to volunteer. I'd be very happy to hear suggestions.
Very nice.
Hey, good to
Yes,
see you.
good to see you too.
Really?
Stay safe.
So, yeah, I thought I'd come to see.